Hi guys, this is Mrs. Foy, and this is part 1 of the Cellular Respiration lecture. So this is for AP Biology for Chapter 9 in the Campbell Biology book that talks about cellular respiration. So guys, we are getting down in the nitty-gritty. We're taking a deep dive into the biochemistry of what's going on in one of the most important chemical reactions for life on Earth. And so it is really important that you take good notes. If you are overwhelmed, you can go back and look at some of the big picture slides to sometimes we get lost in the details and you kind of forget what you're trying to understand. And so I'll try to help you as best I can do that. But be prepared. Be prepared for a deep dive. We are going to learn today how this chemical reaction can take the potential energy from the bonds in food and can make some smaller usable molecules that we use for our everyday energy that we call ATP. And the great majority of life on Earth does this, of course, including plants. And so hopefully we're going to get a picture of that today. So I want to start off with where we are. All right, so when we talk about cellular respiration, we are talking about for organisms that are eukaryotic, organisms that have a nucleus and have organelles. We are talking about a 3-part chemical reaction. And we're talking about something that happens in the mitochondria. So remember when you were baby freshmen and you learned that the mitochondria was the powerhouse of the cell, right? So now we're going to learn about why that is. So what is going to happen here is that this grand chemical reaction that is going to take place is going to actually be broken down into 3 parts: glycolysis, Krebs cycle, or the citric acid cycle, and something called the electron transport chain that happens at the end. And each of these stages are going to produce some ATPs for us. But I want you to think of this as a 3-act play. And in today's lecture, this is just part 1, I'm going to talk about glycolysis and Krebs cycle. And then I'm going to leave the electron transport system for part 2, and I'm also going to talk about what happens when we don't have oxygen present. So I love this. Life is work, right? Totally. But living cells are going to require energy for them to be able to do the work that keeps our cells functioning. And for the great majority of life on Earth, this comes from the chemical bonds that is stored in food. Now, if we were plants, we would be making our own food. And so they have an advantage, right? They're making their own food and then they burn that food. But for those of us that are heterotrophs, this is why I actually start with cellular respiration. We do photosynthesis later. It's because this is what we do. Okay, we are more knowledgeable about cellular respiration than we are photosynthesis, but just remember that plants do cellular respiration too. So we have, if we were going to eat this apple, all right, that apple has some glycogen in it, some starches. It also has some sugars, but this is an exergonic reaction. Energy out, remember, and we're going to when we actually burn the fuel that is our food, we take some of that energy and we use it to do an endergonic reaction, which is the reaction where we take ADP plus a phosphate and make some ATP. And in that process, we use oxygen and we produce CO2, water, and some ATPs. So this slide is a really important slide because this shows you kind of the big picture about what is happening on both sides. So, and it's showing a plant, right, which does both. So I'm going to do photosynthesis over here. So we have the sun's energy and carbon dioxide and water, and that forms glucose and oxygen as a waste product. And then in cellular respiration, we have the mirror image of that. So we have glucose, and we react that with oxygen, and we make carbon dioxide, water, and some ATP energy. So you can see that the reactants of one of these great reactions, this is photosynthesis, the reactants of one are the products of the other. And the products of the other are the reactants of the other reaction. And so let's see if we can figure out what this picture is showing here. So here I've got sunlight energy coming in, and I've got carbon dioxide. Notice that carbon dioxide is coming through into the leaves. And then I have water. Where's water? Well, water is down here in the roots. A lot of people think that that the water is absorbed in the leaves. It's not. It's absorbed through the roots, and it's transported up into kind of a circulatory system that the plant has. And in the leaves, in the chloroplast to be specific, is where photosynthesis occurs, and that's where glucose is made. So here's some sugar that's made right here. And the other product that is made is oxygen. So here's molecular oxygen, O2, that's coming out of the plant. And so what the plant would then do with that glucose is in the mitochondria of the cells of these leaves, it would burn that glucose in the presence of oxygen, and it would produce carbon dioxide and water and some ATP energy that the cell would use to do what it needs to do to stay alive. So a question that I used to like to ask my students for an essay question is I would say, you have a healthy plant and some nice nutrient soil, and you put that plant in a sealed container, okay? And it's sealed, but it's you keep the temperature at a nice, comfortable temperature, and you put it by a sunny window. How long would the plant survive? And the answer is, of course, the plant would survive a very long time, because what it needs in photosynthesis, it is making in cellular respiration. And what it needs for cellular respiration, it is making in photosynthesis. So it's recycling. Do you guys see? It's this beautiful cycle. What is the only thing that is not cycled? The input of the sun, right? So as long as you had sun, you could keep the system going for a long time. So this is a little cleaner picture of what I just said, right? So it shows that our carbon dioxide, water, and oxygen is recycled from the chloroplast to the mitochondria. And of course, if you're in a plant cell, you have both of these. If you're in an animal cell, we just are going to have the mitochondria. But what does not cycle is energy, right? So molecules recycle, but energy does not. So the energy is coming from the sun, and the energy gets fixed into these organic molecules like glucose, which then are burned in a chemical reaction that produces some ATP. We use ATP to do most of our cellular work, and in that transfer, we lose a bit of energy that is lost as heat. And that ends up going out into the universe and increasing the entropy of the universe. And so that is why this system continues to work. So energy is a one-way transfer, but the molecules are recycled. So one of the things that is the important thing to know here is to keep in mind that try to keep in mind these four things: oxygen, carbon dioxide, water, and glucose. Okay? If you can kind of keep track of those four things, you'll be in good shape. All right, so you remember, if we haven't already done our POGL activity on cellular respiration, we soon will. And this gives us a great setting of the 3-act plays. So cellular respiration is a play in 3 acts. Act 1 is glycolysis. Act 2 is the Krebs cycle, or also called the citric acid cycle. And yes, I'm very sorry, but you have to know both names. And then the final finale is what we call the electron transport chain. And the electron transport chain is where we make a whole bunch of ATPs. So let's first take a look at where these things are. Glycolysis is occurring in the cytoplasm of the cell, right? So I'm in the cytoplasm of the cell, okay? And then the product that it makes are 2 molecules of a chemical called pyruvate, and that goes through a little tiny reaction we call the link reaction, but then it ends up going into the mitochondria. So this thing right here is the mitochondria. And the mitochondria has 2 major parts. There's something called the matrix of the mitochondria, which is the gel-like substance in the center, so to speak. And that's where the citric acid cycle occurs, or Krebs cycle. But then on the inner membrane of this mitochondria. So a mitochondria, remember, is one of those organelles that has a double membrane. And that's really important, okay? That's a lot of surface area for something to happen. And on the inner membrane, this is where the final act, the electron transport chain, and we abbreviate that as the ETC, is going to occur on that inner membrane. And this is where we're going to get a whole bunch of mitochondria that are made. And here's my overall equation. And you might say, what the heck are these, Mrs. Foy? Well, these are some coenzymes that we're going to learn about that are going to transfer some electrons for us. Because after all, this is all about the chemistry. So we're going to learn about that. So here is another diagram that shows me all 3 acts. So let's see if we can take a look at this, right? So here I have glycolysis that is occurring in the cytoplasm of the cell. That happens first. And you can see, I like this picture because it shows the whole thing. I have 1 glucose molecule. So if I start with 1 glucose molecule, I end up making 2 pyruvates. And I'm going to show you how you can keep track of that. Those 2 pyruvates are going to go into the matrix of the mitochondria. Whoops, spelled matrix on. Matrix of the mitochondria. Matrix of the mitochondria, and it's going to go through this chemical cycle called Krebs cycle, or the citric acid cycle, but it's kind of like double, right? Because this pyruvate goes through this one, and this pyruvate goes through this one, and I make some carbon dioxide, NADH, FADH, ATPs. We'll talk about all that later. And then it's going to go to the final stage, which is the inner membrane of the mitochondria, inner membrane of the mitochondria. And on that inner membrane of the mitochondria, we have the last act, the electron transport chain. And in the electron transport chain, it's exactly what it says. Electrons are being transported in a chain. And these electrons are going from high energy electrons down to low energy electrons. And every single reaction, it's a series of little waterfalls. Each one of these you can see has a negative delta G, so they're spontaneous. And each one of these is going to result in some ATPs being made. And at the very, very end, we have oxygen, right? So this, number 1, is glycolysis. Number 2 is this little thing called the link reaction that gives rise to the citric acid cycle, or Krebs cycle. And then number 4 is our ETC. So one of the things that I think confuses students, I know it confused me, is I always thought that you had to start with glucose, right? I always thought that, well, you had to start with glucose and then you had oxygen, and it makes CO2 and water and some ATPs, okay? But that's a little bit of a simplification. We don't always start with glucose because the fact of the matter is that you can get ATPs from proteins, from polysaccharides, or from fats, okay? And so one of the things that we're going to see here is that we're going to see in these catabolic pathways, right? Catabolic pathways means breaking down bigger molecules into smaller ones. That's what we do when we eat our food. We're going to get some energy because it doesn't always have to start with glucose. So you can see that some of these breakdown pathways end up making something called acetyl-CoA. And the acetyl-CoA is going to fit into kind of act 2 of our cellular respiration and is going to make some ATPs. So it doesn't always have to be glucose, so just kind of keep that in mind. But one of the things that we're going to clearly see is there is a big difference in the way ATP is made, guys. ATP can be made aerobically. That means oxygen is required. Or ATP can be made anaerobically with no oxygen. And some of those pathways have some things in common. So they both require glucose and an investment of 2 ATPs. And they both are different ways of generating, I almost said electricity, generating energy, okay? But the aerobic process, if we have oxygen present, we can make a whole lot more ATPs. It's performed by eukaryotic cells, and it uses oxygen as the final electron acceptor, because we're going to see this has to do with electrons, okay? Anaerobic respiration for most organisms we call fermentation. And there is a difference, but I'll talk about that in my next lecture. And so it's not very efficient. It only produces ATPs, but you don't need to use oxygen. And it produces some other products which we'll be talking about. Yeast cells perform this, some bacteria perform this, and actually our muscles can perform this for a short period of time. So, cellular respiration includes both aerobic and anaerobic steps, but usually when we talk about cellular respiration, we talk about aerobic respiration, and when we're talking about anaerobic, we're talking about fermentation, okay? And so, even though, as I just said, you can use carbohydrates, fats, and protein as fuel, we usually think of it as starting with glucose. So, here is the balanced equation. Glucose plus 6 oxygen yields 6 CO2, 6 water, and energy. And actually that ATP should go over here. So the energy is in the form of ATP and of course heat, because every time we transfer energy from the chemical energy that is in the glucose gets transferred to the chemical energy in the ATP, I'm going to lose some heat, right? I'm going to lose some energy because of the second law of thermodynamics. So you don't have to memorize this, but I just want you to see this, right? So here's food, right? I don't know what you had for breakfast this morning. I had granola, milk, and some nuts and some fruit. And so that includes polysaccharides, fats, and proteins. So right now, my body is digesting these bigger molecules into some smaller ones. And so I can use amino acids as fuel. I can use fatty acids as a fuel that ends up breaking down into this molecule called acetyl-CoA. And it also can be transformed into it can actually go right into glycolysis. Okay, and some amino acids can go into glycolysis. So there's lots of different places that that the different food pieces, after they're broken down by digestion, can fit into the 3 acts, right? So here's glycolysis, Act 1. Here's Krebs cycle, or the citric acid cycle, and acetyl-CoA, the link reaction. We call this the link reaction in Act 2. And then finally we get Act 3 in our electron transport chain. And I make a few ATPs here, a few ATP here, but a lot, the mother load of ATPs are going to go in my electron chain. So guys, I need to dive a little deeper into the chemistry. And if you've had AP Chem, then you already know this, which is awesome. But guys, there are some types of chemical reactions that involve the transfer of electrons. And we call these oxidation reduction reactions, or redox reactions for short. They always come in pairs. In oxidation, the substance loses electrons. So if something is oxidized, it loses electrons. Well, the electrons have got to go somewhere, right? And the electrons do. They go to the other substance that then gains the electrons, and we call that reduction. Okay? Because it reduces the positive charge. All right? So that's kind of hard to remember. But I have another way that will help you. And here it is, the famous Leo the lion goes grr. If you remember this, Leo the lion goes grr, you will remember what the heck is happening to the electrons. Leo stands for loss of electrons is oxidation. And if you remember that, you don't even have to remember grr, but it stands for gain of electrons is reduction. So remember, Leo the lion goes grr. Loss of electrons is oxidation. So here's a very simple little chemical reaction that shows us a redox reaction. So here is sodium. What happens to sodium? Sodium loses an electron and becomes sodium ion. Here is chlorine. Chlorine gains that electron that the sodium lost. So here's and it would form, of course, sodium chloride, the ionic substance that we know as salt. But this is what is happening when we say that glucose is burned. Okay, you can't say that anymore because now you're a grown-up AP Bio student, and technically what we're saying is we're saying the glucose is oxidized. Okay, so oxidation is what is technically happening to the glucose. And what is oxidation? Loss of electrons. Whoops. Leo. Loss of electrons is oxidation. So what is going to happen to my glucose? It's going to lose electrons. And so what is going to happen is my glucose is going to become oxidized. It's going to lose electrons and become carbon dioxide. If the glucose is oxidized, right, this is oxidized, then something else has to be reduced, and you're right, and it's oxygen. Oxygen gains those electrons, oxygen gains the electrons, and it becomes reduced, and it becomes water. Okay? Now here's something that's just going to blow your mind. Remember, oxygen, Lord of mercy, oxygen is reduced. Oxygen is reduced. But anything that is reduced is itself called an oxidizing agent. So this is, if you might think, well, why isn't oxygen oxidized? Oxygen is an oxidizing agent. It is itself reduced. So anything that's reduced we call an oxidizing agent. Anything that is oxidized we call a reducing agent. All right? So I got a great tip for you guys. And this is, if you're a chemistry major or a chemist and you're hearing this, you're probably rolling your eyes. But I don't care. Because in biological systems, we have a nice little shortcut. We follow the hydrogen atom to see what has been oxidized and what has been reduced. So wake up if you're falling asleep, because this is really important. Electrons will follow hydrogen protons. Where are hydrogen protons? They're all over the place. They're ubiquitous in living systems because we're always in a water environment. So in order to see where the electrons go, right, what is reduced and what is oxidized, follow the hydrogens. All right, so let's look for a second. here is glucose. What is the difference between glucose and CO2? I could ask my grandson that question, and he's in 5th grade. He'd be able to tell you. There's no hydrogen there, Nana, he would say. So the electrons follow the hydrogens. So I can see that I've lost hydrogens, and that is a cheap way for me to see that I've also lost electrons. Well, where did they go? Well, let's look here. What is the difference between oxygen and water? And again, my 5th grade grandson will say, well, Nana, the oxygen doesn't have an H, but the water does. So those electrons got dumped onto the oxygens, the oxygen, and the hydrogen protons followed the electrons, and I see hydrogen showing up. So that is a way that I can see, because the oxygen gained hydrogen to become water, I can see it's reduced because the electrons are followed by those hydrogen protons. So let's see if it works with something else. So here's another fuel. Here's methane. Well, we don't eat methane, but methane is a hydrocarbon fuel, just like glucose is, and so the same type of reaction is going to occur. Look what happens here. I have an H here. Over here, I don't, right? So what has happened is that the electrons have been lost they've been stripped off the methane, and the oxygen is going to take those electrons, right? They gain those electrons and they dump them on, it's been dumped on that oxygen, and the hydrogen protons are added that actually came off of here. And that's how I get water, okay? So the methane is oxidized and the oxygen is reduced. And remember, oxygen is an oxidizing agent. It itself is reduced, and I can see this because I'm following the hydrogens to see where the electrons go. So it's all about the electrons. I'll boil it down to very simple. I've got a bunch of electrons in the carbon to hydrogen bonds of the glucose molecule. I'm going to basically strip off those electrons in a bunch of different chemical reactions. And it's not until the final act of cellular respiration in the electron transport chain that I'm going to see the fruits of my labor. These ripped off electrons in the oxidation, right? Loss of electrons. In the oxidation of glucose, those electrons are going to be passed down in a series of small redox reactions. That means somebody's losing electrons, but then somebody's gaining. And every single time I do this, enough energy is siphoned off those electrons to end up making me some ATPs. At the very end of this, I got to do something with these electrons. Notice they're very low energy electrons, and it just so happens that oxygen is right there to catch them. Oxygen, as you know, if you've had chemistry, is extremely electronegative. In other words, it is an electron magnet. And so oxygen becomes reduced. It takes those electrons, and look, there's my two hydrogens that are always around, and I end up making water. So that's going to be the final act of my cellular respiration process. So where are these electrons? We can't just have naked electrons floating around, okay? That is a bad situation in living things. So these electrons are going to go on something called a coenzyme. Sometimes we call these electron carriers. And it is a molecule called nicotinamide adenine dinucleotide. Luckily for us, we can shortcut that and call it NAD. NAD is a coenzyme. It's an electron acceptor. All right? It's an electron acceptor, so it will be reduced. Well, I'm going to guess that I'm going to see that it's reduced. It's going to have a hydrogen on it. And that's exactly right. When NADH is reduced, there are some hydrogen protons that are around there, and I see NADH, okay? So NADH is the reduced form of NAD. And why is that important? Because there's electrons on there. And remember, I told you that the electrons have energy in them. And so those electrons are going to come into play later when I want to make some ATPs. So here's a little cartoon that's going to help me realize that, right? So here's my organic molecule. Like I could think of this as glucose, or I could think of this as some other food that I'm using as fuel. And so what you're going to see in just a minute when I explain this is you're going to see some electrons being ripped off and temporarily being carried on this NAD coenzyme. It itself is going to be reduced. Okay, so the organic fuel is going to be oxidized, right? So it lost its electrons, and the electrons are dumped onto this NAD, which becomes reduced and forms NADH. A lot of times they'll write it as NADH plus H, and I don't really care that you know that. I just want you to know that it's been reduced, and we usually write it as NADH. Okay? So here's the actual molecule, and no, you don't need to know this. You don't need to memorize this for this class, but let's just look and see if we can see the difference. Here's the oxidized NAD+, here's the reduced NADH. Do you see a difference? Right here it is, right? Exact same molecule, but this has another hydrogen on it. And remember, the hydrogen is what I'm looking at, because that is where the electrons are, right? The hydrogens are just a cheap way to see where the electrons went. But I'll take it. I'll take it because it's easy to see. So we're going to see this NAD acting as an electron shuttle, right? It's this coenzyme, this electron carrier, whatever you want to call it. It's going to get electrons on it, and then it's going to be NADH. And then I'm going to use these electrons. They're going to be shuttled someplace further down the line, and eventually I'm going to use it to make some ATPs. It should make sense to you that once I make some NADH, I'm going to have to figure out a way to oxidize that again. This is reduction. But I'm going to need to oxidize that later on to get it back to NAD. Otherwise, everything's going to go to a screeching halt, right? So I do have, this is a reaction that can be reversible, and there's an enzyme that makes that happen. You don't have to know that, but there's an enzyme for each one of these. So these electrons that are going to be ripped off my food, if I just did that in one big step instead of a bunch of little ones, I would have an explosive reaction. But that would waste a lot of energy. And so what our cells do is they do this in tiny little steps, which we'll see. But at the very end of the day, what happens to those electrons? And it's oxygen. Oxygen is the final electron acceptor. Final electron acceptor is oxygen. And oxygen, I tell my students, oxygen is like the catcher waiting at the end of this hot potato electron acceptor, electrons being passed. Oxygen is going to accept, and when it accepts, the hydrogen protons are always around, right? And it's going to make water. And those electrons are going to be used to ATPs. So here is back to this this free energy thing, right? So I've got some high energy electrons that are up here. If I let the hydrogens react with the oxygens in one big step to make water, it would be an explosive release of heat and light. And so instead of that happening, we're going to see that the electrons are going to be passed in a step-by-step process, kind of like electron hot potato. That's what I call the electron transport chain. Electron hot potato in the electron transport chain. And I am going to siphon off enough energy to make some ATPs, but still the same thing happens. I'm still making water. I still have these electrons, but now they're lower energy because they've gone down a series of transport. Oxygen is still going to accept the electron, and I'm going to make water. All right, guys, get ready. Here we go. That was all the background. Now we should be able to proceed pretty quickly. Remember, there's 3 acts in my plate: glycolysis, citric acid cycle, also called Krebs cycle, and the ATP synthesis, which is called the electron transport chain. So one thing I need to tell you is that I'm going to phosphorylate. The whole idea is that I'm going to take some ADP and I'm going to phosphorylate it. But that has a positive delta G. So it's not going to happen spontaneously. I need some energy, and the energy is going to come from those electrons to make some ATPs. And so oxidative phosphorylation is when I do this process that oxygen is required. I am going to make some ATPs in a different way in glycolysis and in the Krebs cycle, but it's not using oxygen. All right? And I've tried to color code the rest of this lecture for you and part 2. Anything that has to do with glycolysis, I've put in a blue background. Anything that has to do with Krebs cycle or the citric acid cycle, I made yellow. And then the next lecture, I'll talk about the electron transport chain, which I made green, just so that you can kind of keep track of where we are. So let's look at the place where we are, all right? This is my cell, okay? So this whole thing right here is my cell, right? And glycolysis is happening in the cytoplasm. So the first thing that's going to happen is glycolysis is going to be broken into pyruvate. Glucose has 6 carbons, and pyruvate has 3 carbons. 3 carbons, but there are 2 of them. So one of the things you're going to hear me say is count the carbons. That's really important to count. So those two pyruvates are going to go into the citric acid cycle, also called Krebs cycle. And then finally, we're going to get into the electron transport chain that's on this little inner membrane here. This is happening in the matrix. Okay? But I told you this all has to do with electrons, right? So I'm stripping electrons from my fuel, and the electrons are going to be shuttled to the electron transport chain in glycolysis and also in the Krebs cycle. And you're going to see NADHs and also a cousin coenzyme that also is an electron carrier called FADH2 that can also carry some high energy electrons for me. And you see here that I am going to make some ATPs, but the way that I make ATPs in glycolysis and citric acid cycle is called substrate-level phosphorylation. And substrate-level phosphorylation is a fancy way of saying you take a phosphate, you plunk it on an ADP. There's not a fancy way of making it like you are going to see in oxidative phosphorylation, which we see in the electron transport chain. So stay tuned. There are different ways to make ATP, and you need to know the difference between substrate-level phosphorylation and oxidative phosphorylation, which happens in the electron transport chain. Oxidative phosphorylation accounts for 90% of the ATPs that are generated from my fuel. I have a few little ATPs, 10% of them, that are produced by substrate-level phosphorylation. All right, guys, here we go. So we're going to go through this pretty quickly. You can see that this is blue, right? So I must be talking about glycolysis. And we're going to look at this in general, and then I'm actually going to look at each individual step. But good news, don't fall off your chair. You're not going to have to memorize this, okay? So we're going to see glucose. This literally means glucose splitting, glycolysis, the splitting of sugar into 2 molecules of pyruvate. Count carbons. There are 6 carbons in glucose, and there are 3 carbons in pyruvate. And because I have 2 of them, I get 6, all right? And so the glycolysis occurs in the cytoplasm and has 2 major phases. I invest some ATPs in the first phase, and then I make some ATPs in the second phase. So here we go. This is kind of an accounting sheet. In the first phase, I'm going to use up some ATPs. And you might think, well, why in the heck would you? I thought the whole point was to make ATPs, Mrs. Foy. This is true. But I have to use some ATPs in order to be able to make more. And so I'm going to use up some ATPs, and then you're going to see that I actually make some. I use 2, and I make 4. And so I end up with a net overall of 2 ATPs. At the end of glycolysis, I'm going to have 2 pyruvates. And these pyruvates still have some high energy electrons in them. All right, so don't be sad because we've still got some more energy that is left there. And I am going to split off some electrons, right? So these electrons that I rip off my glucose are going to go on to my electron carrier or my coenzyme NAD to make some NADH. Okay, so remember there's electrons on there that I am going to be able to use to make some ATP. All right, so guys, I'm going to go very quickly through the steps of glycolysis. What did I say? You do not need to memorize. Okay, you do not need to memorize this. I'm just going to show you the steps to just kind of familiarize with to familiarize you with the steps, but you do not have to memorize this. If you take biochemistry in college, and you know when you go to med school or nursing school, yeah, you're going to have to know all these steps, but not for AP Bio. So here we go. First thing I do with glucose is I'm going to use some ATPs, right? Remember, this is the investment part, and this always shows you the enzyme. So the enzyme is hexokinase. I make something called glucose 6-phosphate. That glucose 6-phosphate gets a little shuttling around. I make an isomer of it called fructose 6-phosphate. And that's just moving this phosphate group. Remember, when you phosphorylate something, you make it a little more unstable. When you make it more unstable, you make it more likely to react, right? You make it more likely to react. And so that's why the cell is the cell is doing this. It's phosphorylating, so it's more able to react. So now look what I'm doing. I am using another ATP. Oh, for goodness sake. I thought I was supposed to be making ATPs. I'm using ATPs. This is true. But I do want to point out to you this enzyme, phosphofructokinase. Will you write a star in that in your notes? Because phosphofructokinase is very important for us to keep track of later. It's going to help us to regulate glycolysis. So just make a little note that that is one enzyme in glycolysis that I do want you to know. So anyway, I make this thing called fructose 1,6-bisphosphate. And so now I'm going to shuttle around a little more. I'm going to make something called glyceraldehyde 3-phosphate. And so at this point, I'm going to strip off some electrons. So how do I know that? Because I see this NAD goes to NADH. That means that the electrons got stripped off this glyceraldehyde 6-phosphate and got put on my NADH. And I've got some phosphates there that I'm going to plunk on there. And here's where I'm going to make some ATPs. I'm going to split off some of these molecules, and I'm going to make 2 ATPs and some more little shuttling that happens here, some more shuttling, rearranging, and finally I'm going to end up making 2 more ATPs and a molecule called pyruvate. Pyruvate has 1, 2, 3 carbons, all right? And so this is done. I'm done with glycolysis now. What has been the result of all my hard work? I made 4 ATPs, I made 2 NADHs, and I made 2 pyruvates, all right? And that's really important. So now I'm going to go into Act 2. Hang in there. We're almost done, all right? So this is Act 2 of my cellular respiration play. And now I'm going to start with pyruvate, okay? So far, everything that happened in Act 1 was in the cytoplasm, right? So Act 1, which was glycolysis, is going to happen in the cytoplasm of the cell. Now we're going to move to Act 2, and this is going to happen in the mitochondria, specifically in the matrix of the mitochondria. In the matrix of the mitochondria. So what's going to happen is that everything is going to come to a halt unless I have oxygen. I must have oxygen to proceed into the citric acid cycle. If not, fermentation is going to occur, and I'll talk about that in part 2 of my lecture. So the first thing that's going to happen is that you have to remember I have 2 of these pyruvates, okay? I'm only going to show you one, but you have to remember you're going to do all this times 2. And so the first thing that's going to happen is I'm going to take pyruvate and I'm going to convert it into acetyl-CoA. This is sometimes called the link reaction because it's linking glycolysis with the citric acid cycle or Krebs cycle. Okay? And here's what's going to happen. Here's my 3 carbons, right, of pyruvate. Well, the first thing that's going to happen is carbon dioxide comes off. And you're like, Mrs. Foy, that was one of the 4 molecules that you told me to keep track of, remember? Okay, I told you to keep track of these. We've already talked about glucose. Now we're starting to see carbon dioxide. And we're going to see more carbon dioxides in just a minute. So I have a carbon dioxide being pulled off of here. And that means I'm going to have 2 carbons left. Here, I'm stripping off some electrons, right? The high energy electrons from the pyruvate are going on to my NADH, going from my NAD to my NADH, right? And this is going to go to the electron transport chain, right? So it's going to shuttle those electrons. We'll see that later. And then there is this molecule called coenzyme A that gets plugged onto what was left of my pyruvate. So here's the 2 little baby carbons that were left from my pyruvate, which of course started from a 6-carbon glucose. At this point, this is now going to produce a bunch of things for me. So 1 pyruvate is going to produce 1 ATP, 3 NADHs, and 1 FADH2 per turn. And remember, this is going to be times 2 because I make 2 pyruvates per glucose. And this is all happening in the matrix. Okay, so again, you do not have to memorize. You do not have to memorize each of these steps. Okay, you do not. But you do need to know the 3 basic stages. Okay, so let's look at that. So here I've got the link reaction. This is the link reaction. And look, here are my carbons for me. Here's 3 carbon, here's 2 carbons. So at this point, this is the link reaction. And at this point, acetyl-CoA with its 2 carbons is going to go into the citric acid cycle. And the citric acid cycle is going to have 3 basic steps that are happening. I'm going to produce some more carbon dioxide. Right? So here's 1 carbon dioxide, here's 2 carbon dioxide, and if you're counting carbons, you're like, well, Mrs. Foy, I am now done with all the carbons that came from the original glucose, and you're right. But the cell has a way of fixing that so it can keep going in a cycle, and I'll explain that in just a second. The other thing that's being produced is I've got electrons being stripped off what was the pyruvate, which became the acetyl-CoA, and those get bonded on to the NADH. Remember, that's going to go to the electron transport chain in step 3. I'm going to make a couple of ATPs by substrate-level phosphorylation. This is just like the way that I made them in glycolysis, substrate-level phosphorylation. And I'm also going to strip off some other electrons and put it on this cousin electron carrier called FAD, which gets reduced to FADH2. All right, I think I said all of that. So now we're ready to look. Okay, again, do not memorize each step. But I do want you to be able to recognize the stage. So you need to recognize the stage. All right, so here we go. Acetyl-CoA. First thing that happens is acetyl-CoA is going to be bonded to a 4-carbon molecule called oxaloacetate. Oxaloacetate plus the 2 carbons on my acetyl-CoA make a 6 carbon molecule called citrate, which is why we call it the citric acid cycle. I should have said it's also called Krebs cycle because a dude named Krebs got credit for discovering this, okay? So we call it both, citric acid and also Krebs. So I get a 6-carbon molecule that's made. That 6-carbon molecule gets rearranged. I get some water that's being taken off. And now look what's happening. You should recognize this. I get some electrons that are stripped off, and I get some NADH to be produced. That's going to go to my electron transport chain. But look, I have some more carbon dioxide being given off, and that's something I need to keep track of. So now I've got 1, 2, 3, 4, 5 carbons that are left from my 6-carbon. Look. Here is another carbon dioxide being given off and some other electrons, high-energy electrons, being stripped off. And I make this molecule that is now 1, 2, 3, 4 carbons, okay? And this is succinyl-CoA. So my NADHs are going to go to my electron transport chain, and my carbon dioxides are going to be released as waste, okay? Now I'm going to make some ATP, and this ATP is not the fancy way I'm going to make it in the electron transport chain. This is called substrate-level phosphorylation. Substrate-level phosphorylation. I'm going to make some ATPs, and then I'm going to strip off some other electrons to go on this FADH2 that will also go to the electron transport chain. Notice I still have 4 carbons, 1, 2, 3, 4, and I go from this molecule called fumarate to a molecule called malate, 1, 2, 3, 4, and then my final stage is I strip off some more final energy, some electrons that are higher energy, and I have some more FADH, and I still have my 4-carbon molecule oxaloacetate. So all of this is happening once I get the CO2s gone here, and remember there was another CO2 that came off in the link reaction. I basically have chopped up my 6-carbon glucose and I'm done with it. But the reason why I have this 4-carbon molecule of oxaloacetate is so I can make this a cycle that keeps going. And that's really important. The cell likes cycles because they're very efficient. What did I make besides CO2? I made a bunch of NADHs and FADH2s, and I made an ATP. But remember, this is going to go twice through, so I actually make 2 ATPs. That, guys, is the end of part 1, all right? And so in part 2, we're going to talk about what are we going to do with these NADHs. What are we going to do with this FADH2? And what are we going to do with these electrons? And how are we going to make ATPs with oxygen? So stay tuned to that for part 2. And I hope this has been helpful. I'll see you in class.